So our next presenter hasn't had to travel too far because he's presenting in his hometown. Most of us get on a flight and have a bit of fun, but there you go. So Gildas Hounmanou is going to present on a decade of Vibrio and Chikungunya infections in Denmark, and there's a correlation with high seawater temperature and genome-wide association with environmental isolates. So the floor is yours. Thank you.
Thank you. And welcome to the show. I'm taking you to a kind of nasty road. You could see that already on the screen. And I'm sure most of you, in the next 2 months, you're likely going to join, to go back to the sea in your beautiful bikinis, or some of us in the beautiful swimming suits, because we love going to the beach. But then we need to be aware that it's not always safe out there. And that depends on who we are, right? So we have noticed over the past decades or centuries that there is an increase in the sea level, seawater temperature. And that is mostly seen here along the Baltic Sea, where we've noticed a very more than 1 degree elevation in the seawater temperature. And this becomes an issue mostly in the summertime. And we also know at the same time that as the sea surface temperature rises or the ambient temperature rises, humans are likely to go swimming. And we see these kind of behaviors mostly in the summertime, and that is what we do, and that's what we love to do. And it's nice to go to the beach, and you should go. I'm not scaring you away. But you should be aware that over the past decades, we have started seeing a lot of cases of seawater infections, mainly caused by Vibrio species or some Chikungunya species. And these have become very, very common, especially in the Nordic countries. Both along the Baltic Sea, but also on the other side, even in Germany. So you need to be aware of this, and this is what we went to look back into, especially through the Danish data. The data I'm showing next is from the past decades, but data from back all the way from 2020 all the way to 2010 to 2018. And what we see is that there is a very strong correlation when you look at the number of cases, the incidence of Vibrio and Chikungunya infections, when you compare that to the variation in seawater temperature. And these strong correlations are, again, evidence that whenever there is a very high temperature during the summer, like when you have warmer summers, you're likely to have more of these kind of infections in the clinic, and the doctors are kind of aware of this. As this goes on, it happens mostly in the summertime. As I said, you could see the peak goes off mostly around the week 21st, all the way to 30 and 35. Those are the periods or the seasons corresponding to the summertime, and that's where you have most of the cases. They seem to occur mostly in the younger ones, but also in the elderly. And there is a reason for that. We all know that in the elderly population, there is an immunity issue. But also people with some liver conditions are likely to be more at risk of these kind of infections. And also, if you're going to swim in the summertime along the Baltic Sea, you are also advised to cover your wounds if you have some open cuts, so you avoid these kind of infections. And you're also likely to have very severe cases if you are having liver diseases. You could end up in septicemia cases. Most of the species in cause here, they are quite varied. Most of them are Vibrios, some are Chikungunya. And according to the type of infection you get, of course, I forgot to mention the ear infections, which are quite high here. And according to the type of infection, there are some species that seems to be associated with some type of infections. And Vibrio vulnificus, which we call here mostly like the Baltic germ, seems to be the one causing most of the septicemia cases. And you can see the septicemia cases are not much here in this graph, but I'm going to emphasize on it because these are the ones that cause most of the death. And the wound infections are usually the ones that end up with amputations, and you don't want to swim and then the next year you don't have your arm anymore. So, when you look at the vulnificus cases that end up in septicemia, they have a fatality rate that is very near the Ebola septicemia risk, which is like 50%. So that is quite high. And most of the cases here, this is the Danish map. We have found most cases as we move down towards the more brackish area of the sea, because the North Sea, as you could see, is more saline, and those who do oceanography know that the North Sea is quite more saline than the Baltic Sea. And maybe you don't have these kind of cases also along the Mediterranean Sea or the Atlantic Sea, simply because they are quite higher in salinity. So as you move towards the areas where the Baltic Sea is less saline, you're likely to see more cases. And also sometimes in the summer camps where people are likely to go in summer vacations, you have more of these cases here. So all these data have been recently published just last month in Emerging Infectious Diseases. And you could go online and read. It's a very short and nice paper. Thank you. Now, while we're doing that, I went through the SMID research grant process, and I got some money from SMID to do some fun work. And throughout last summer, I was, while people were enjoying the sunshine, I was kind of walking around taking samples through all these sites at the Baltic coast, from Ulan all the way down to Shilan. And I analyzed these samples in the lab. And you could see I'm trying to show here the kind of prevalence of different species that I recovered from the summer compared to what I have in the fall and also down in the winter time. And clearly, if you look, the way you read it is look at this key up here, where from blue to red is like you have more cases. So we seem to be having more cases in the summertime, more samples positive in the summertime, sorry. But again, this is species dependent. And the species that I'm much interested in is this Vibrio falnificus. Of course, you don't have much of it in the winter or in the fall, but you have it growing much more in the summertime, which you also see here for other Vibrio species. Now, diving deeper into the Baltic germ, Vibrio falnificus, I sequenced them together with some clinical isolates from 2018. So these are environmental isolates from mussels and seawater, seaweed, and also isolates from the clinical samples. So they're all sequenced, and you could see first, if you look at them, MLST is more like super heterogeneous samples. But when you kind of go down to run a phylogenetic analysis, we start seeing a bit of clustering here. First, these samples came, the clinical samples, are red. They came from Finland, Sweden, Norway, and also in Denmark. All the red dots are the clinical samples, and the yellow and blue are environmental samples. The yellow ones are from mussels, and the blue are from the seawater, right? So what I'm showing here is there is a cluster of mussel samples and clinical isolates here with quite short SNPs, like less than 10. I didn't have the SNP data here. And we also have a very large cluster down here, gathering water isolates and clinical isolates. The whole point that this is telling is that whatever we are having in the clinic is likely coming from the sea. We need to be aware of that. But one issue is that we are not now able to distinguish an environmental sample from a clinical sample when we look at the virulence. And I tried to run a pan-genome analysis on all of them. And you could see the core genome is quite high. It is almost the entire genome of Vibrios. Vibrios, they have like 2 chromosomes, and one is more than 3,000 genes, and the other smaller chromosomes. So looking down into the falnificus pangenome, what you see is that, again, that cluster where you saw the mussel samples together with the clinical isolates, you still see it here with a very, very similar accessory genome as well as core genome. Let's then look into here and see what is happening in the accessory genome is that when you look at virulence genes gathered here by virulence factors, because I cannot just list all the virulence genes here, but I try to put them by virulence factors. And if you move from factor to factor, there is no big difference. This is not statistical information, but I'm just displaying this so you can have this feeling that whether we are looking into a clinical isolate or an environmental isolate, we cannot tell the difference at virulence level, except, of course, for biofilm formation ability, where environmental strains seems to be a bit better in that, and that makes sense because they are environmental isolates. Now, what the question comes now is, we are still unable right now to distinguish or to predict which environmental isolate is likely to cause an outbreak. And this is due to many reasons. One of that is because of the pathogenic mechanism of Vibrio falnificus is not clear. And the other issue is that there is a host factor associated with this. Like if I go swimming and my colleague here also goes swimming, we don't know who gets infected yet. and we don't know which isolate is able to cause infection, but we can see all of them are the same. And we could end up that she got infected, sorry for saying that, or I get infected. And that is likely because there are some host factors on my side that makes it possible. So the next thing I'm doing is, after this grant is over, and I've now submitted this new application to the Danish Research Council, looking into, first of all, the transcriptional pictures. When we have the environmental strain, like when we have environmental changes, through the summer all the way to the next summer, while the temperature varies and the salinity changes and the nutrient content changes, what happens in the transcriptome of the strains? And is it that they just become more prevalent, or is simply because they change virulence mechanism? And are we able now I'm planning to do a huge sequencing work between clinical environmental strains in order to, at the end of the day, be able to tell, to the clinicians or the scientific world that these are markers that you can follow to be able to tell that these are pathogenic strains, and that will be made into a PCR test. We will publish the primers for anyone to use. I'm looking into the host factors. And lastly, which is why I'm very interested in Ben's presentation, I'm developing together with some colleagues in the technical university how best can we use models to predict severity, because that is the issue now in the clinic. Like if you take a person that got exposed to Vibrio vulnificus, we need to be able, based on the strain, the genomic data in the strain, and based on the host's immunity or health data of the patient, we need to be able to tell whether this will be developed into a severe case or not. That is what I'm looking into next. And I would like to thank all my collaborators, Steen and all the others. They are in this room now, Mariana. Of course, ESMID funded the environmental work. SSI, which is the Danish National Institute of Health, most of the data came from them because Denmark has a very beautiful background with data. Thank you very much for listening. I'm ready for any question if we have time.